unsurprisingly, they have a lot of questions about the COVID-19 vaccine development. And I know you're in the thick of that right now. So I guess I sort of wanted to start by asking you to tell us a bit about the approach that your team is taking to it. But just first off, perhaps just introduce yourself and let us know who you are and what you're working on at the moment.
So I'm Colin Pouton. I'm Professor of Pharmaceutical Biology at Monash Institute of Pharmaceutical Sciences. I'm really a drug delivery scientist, but with a broad background in pharmaceutical science. And I have a group that's been working on delivering DNA and RNA for therapeutic applications, or for vaccination. So the opportunity to apply this technology to COVID-19 vaccination is an obvious one for us, and we're quite excited to give it a try. I suppose what's different about the approach we're using is that we're using genetic material, so in this case messenger RNA rather than DNA. Messenger RNA is the molecule which is, if you like, the code for making the protein in a human cell or a mammalian cell. So what we're actually doing is delivering the code. And the advantage of doing that, rather than administering a deactivated virus or a protein vaccine, is that we can change the structure very easily. So we can make molecules of mRNA, change the structure, try different structures and see which ones create the best vaccine quite quickly. And that's why we've been able to, and in fact, the vaccine which is being trialed in the US at the moment, which you probably have heard of, is an mRNA vaccine. Somewhat similar to ours. We don't know what it is exactly because it's a commercial product, but we're using the same technology, and that's the reason it's been so quick. I think the American group that put this vaccine into humans, they actually started the human safety testing 63 days after the genome of the virus was published, which is extraordinary. Normally it takes years for something to get into human safety trials. So we're all learning a lot from this process.
Yeah, it sounds like it.
Yeah.
There are three different sort of candidates at the moment. Is one showing more promise than another?
Well, we're just about to find that out. We don't know which one is likely to be the best at the moment. The reason we've got three rather than just one is really because we can make lots of different candidates quickly using the coding approach, the mRNA approach. And we've got several others in the pipeline as well. The issue really is we don't know at the moment what is the best vaccine target for the COVID-19 virus. We know that it's likely to be the spike protein, which everyone talks about, which is this thing that the reason why the virus is called the coronavirus, this sort of crown of spikes around it, which you often see depicted in the news and so on. That spike is made up of three proteins meshed together, three identical proteins. We call it a trimer. And the structure of that trimeric spike is actually quite complicated. So part of it creates good antibodies. So we raise good antibodies against part of it, and we raise antibodies that are not very useful against other parts of that protein. So in terms of which bit is best, we expect the part which binds to the receptor, the way in which the virus gains entry into the cell, we expect that that'll be the best vaccine. But it needs to be presented in a way that makes it look like the virus. Otherwise, we might be unable to recognize it as our immune system may not raise an antibody which will recognize the virus. So this is where the mRNA approach gives us a lot of scope for testing lots of different structures and seeing which ones are best, much more quickly than if you were doing it by making proteins and trying purifying a protein takes time, and so every protein would be slightly different, and then you have to test them all. So this approach gives us a lot of speed in terms of finding the best candidate. So we're hoping to find that out in the next month or so.
Amazing. What are the impacts of COVID in terms of the work that you're doing? So obviously you've had to change the way you work, speed up this process, but how are the restrictions in terms of social distancing and all those sorts of things impacting on how you're having to do your work day to day?
Well, I guess it isn't having that much effect on the group working on this project. We're allowed to come in and work because we're regarded as essential workers. And so, yes, the social distancing has to happen in the lab, but that's not too difficult. People are often that sort of 1.5 meters away when they're in the lab. The office spaces are actually more difficult than the lab spaces for us. But for the rest of the institute, with doing work which is not necessarily regarded as essential, at the moment they're not able to work. So in a sense, the COVID-19 work is going on as normal. The rest of the university, particularly the PhD students, are somewhat frustrated and hoping to get back in the next few weeks. We'll see.
With the development of these types of vaccines, obviously there are scientists and researchers around the world working at pace. Is there a sense of connection? Is everyone sort of working together to try and speed things up as quickly as possible?
It's, that's a very interesting question. And we're learning more as we go along. It's all happening so quickly that I think to a large extent people are working as at least not individual groups in isolation, but in competitive groups, probably. For us, we definitely need collaborators locally to help us with the virology and handling of the virus and so on. We don't really do that here. So we're working locally with the Doherty Institute at the University of Melbourne, and we'll probably be working with CSIRO at later stages too to test the vaccine in more, if you like, challenge test conditions. Somehow we have to select a vaccine which could potentially go into clinical trials, and that's actually the challenging part. How do you decide which one to use until you don't really know until it's actually in efficacy trials in humans? So you'd ideally like to have some sort of model that you could use that will predict which will work best in humans. And there's a variety of things we can do, but there isn't an absolute challenge test at the moment. Right. So the CSIRO group in Geelong at the Animal Health Laboratory, they do have a challenge model, which we might be able to get access to. But actually, people are selecting vaccines really on the basis of early preclinical data in just looking at what antibodies are raised, and then setting up the permission to do human safety trials. So what we're finding is that there is a bit of a race on, no doubt, around the globe for different types of vaccination. And I suppose what's fascinating about it is there's so much activity. So people are working with their own community, if you like. So we're here in Melbourne working with the Doherty. We don't know too much about the other mRNA vaccines that are in development. Some of them are commercially set in a commercial setting, so we're unlikely to ever find out what they are. So there isn't really that much communication between people working on separate projects, I would say. There is a global organization that is funding a lot of the work, which is a multi-sector organization, so funded by governments, philanthropists, a variety of sources. And that organization is called CEPI, CEPI. It's based in Norway, and they're trying to raise a large sum of money for essentially testing 3 vaccines in a full phase 3 clinical trial.
Wow.
And at the moment, we're all a little bit unsure about which ones are likely to succeed. What CEPI are doing is they're saying we're going to try everything, and we're going to gradually weed out ones that we think are less likely to succeed and probably test 3 of them. But, where it's going in terms of what will be available and when and to whom will it be available is actually very difficult to say at the moment. It's really, we're learning as we go along with this. How you can produce, billions of doses for global use is another huge practical problem.
This sort of brings me to my final question, really. If Monash, for example, and partners happen to be the ones to win the race for the successful vaccine, what are the implications there around IP and the sale of the vaccine versus providing it for free globally?
Yeah, look, that's a really interesting question. And frankly, I don't know. I don't think any of us know who's going to pay and how people are going to provide vaccines for enough people. I think at the moment, what people are focusing on is getting a vaccine which works and then maybe making that available to frontline workers and obviously vulnerable groups, elderly and so on. And I suppose that in the Western world, you have vaccines that are being developed with funders that will supply to communities in the Western world. How that gets rolled out globally in countries that really can't afford to buy vaccines is really, at the moment, very uncertain. I guess, having said that, we do live in an environment now where philanthropists are putting huge amounts of money into public available drugs and vaccines. And so, I think that if a vaccine is developed and is very successful, that initially it'll be tested in those immediate health workers and so on, but probably people will come in and supply globally to developing countries and so on. We're just not at that point yet.
Yeah.
But in terms of IP and, who's going to pay for it, who's going to buy it, it's really quite unknown. So I actually think that it's quite important that Australia has its own source of vaccines, which is one of the reasons why we're working on it, because we realise that there are lots of other approaches going on. But I actually think that if you want to control your destiny in this sort of pandemic, you actually have to make it yourself, because there's no guarantee that we get vaccines from anywhere, really. Some of them are very difficult to make and produce in large numbers of doses. So it all remains to be seen. It's quite fascinating, but challenging.
Yeah, terrific. Professor Colin Pouton, thank you so much for your time today and for answering these questions from our alumni about COVID. And very good luck to you and your team with the progress on the vaccine.
Thank you very much. It's been a pleasure.
Thank you.